Hi, I'm Grace Moon, and this is my project, Temperature's Effect on Photosynthesis in Different Food Crops. This is a continuation on the project I did last year, which was Temperature's Effect on Photosynthesis, in which I used spinach leaves. And I came up with the idea for this project with the help of another person. So I was interning over the summer, and my mentor wanted me to explain my project to the people she was working with. And so she was having me practice with her cousin, who is a food nutritionist and has his own farm and works with crops a lot of the time. So I was telling him this, and he said, "That's very interesting. You know what you should do? You should try what would it be like with different types of crops, because spinach is a cold season crop. What about tropical?" And I thought, that's very intriguing. So that's what I went with. So I thought that spinach would do the best at cold temperatures. The coldest would be its highest. Cilantro would be very stable. It wouldn't really have any spikes because it's a temperate crop, which means it's more suited to neither of the extremes, but kind of both. And banana leaves, I thought that they would have very low transmittance at low temperatures and very high transmittance at high temperatures because they're a tropical plant. And then how I tested this was I made chloroplast solutions out of each of the three types of leaves. So that's basically just blending up the leaves with a sucrose solution and then straining it through some cheesecloth to get the liquids and the smaller parts, including the chloroplasts, of the leaf. And I used that to actually start the photosynthesis. And the way that I synthesized photosynthesis was in different cuvettes, filled with water, phosphate buffer, and DPIP. DPIP acts as a stand-in for NADP in the light cycle, but the difference is that when it receives the H, when it becomes DPIPH, it goes from a solid royal blue color to a very clear, colorless solution. And so that way, you can, if you measure the light transmittance through, you can see how fast the percent transmittance changed or how fast the photosynthesis happened. And so I did that with each of the three types of plants, and I had 2 trials per each. But how did I get them to be different temperatures? Well, I just surrounded them in water of that temperature. So there I had beakers set at different temperatures. There was 9 degrees Celsius, 14 degrees Celsius, 21 degrees Celsius, which was the room temperature of the room I was in, and 28 degrees Celsius, which was the lowest setting on a heat bath. And so these waters were this temperature, and after testing the cuvettes for their percent transmittance, they would spend their in-between time between 0, 10, and 20 minutes of adding the chloroplasts to the cuvettes. They would spend it in that water bath so that they would be that temperature. And now the results from this were pretty interesting. And so first we have the spinach, which was, as you can see, it was pretty consistent and then had a sharp decline. That's pretty consistent with what happened last year as well. That there is a super swift drop off between a room temperature and a above room temperature variable, but besides that, it's pretty consistent. It's doing well at the colder temperatures, which is about what was expected. Then we looked at the cilantro, which turned out that color as well, and like it was thought, it was kind of steady. There wasn't really many spikes. There wasn't anything drastic that was different between the difference in the temperature groups. But there wasn't a gradual increase, which kind of makes sense. If you're a temperate crop, you don't really prefer to be colder because it's probably not as sunny, so you're probably not going to adapt to be that way. So while slight, it did increase that way, and it was pretty steady, so that seemed to fit with what I expected as well. However, the banana leaf group was not quite expected, I would say. I wasn't quite prepared for that. As you can see, it was very low at the low temperatures, but there was not a very high temperature change not temperature change, but at the high temperature change, there was not a high percent transmittance change once it got high into the 28 degrees. Now, the range for the other temperature groups for the spinach and cilantro, the percent transmittance seemed to be from like around 20 to 40 on average. But as you can see, the maximum we had on this group was 12.9, which is not very high. And I was confused for a while. I was like, I wonder what caused this data to be like this. But there are a few explanations. One could be that there are different types of crops, as in cilantro and spinach. They have to be planted every year. They're annuals. So their photosynthesis may be a certain way, but banana leaves are perennials. They come back every year, which means that they may be set up to have a different process of speed for photosynthesis than a plant that is just going to be there and die. It's going to try and live its life and then die versus trying to sustain growth over many years. Also, age affects the rate of photosynthesis that goes on in the leaf. So a new leaf photosynthesizes at a different rate than an old leaf. And seeing how I don't know how old the banana leaf was when it was picked, that was a variable that I had left unaccounted for and could have caused data like that. And just to go over this is comparing the cilantro, the spinach, and the banana leaves at different temperatures. So the spinach was highest out of all of them at the coldest temperature, which makes sense. But then we go to the next temperature up, and they were pretty similar, but the cilantro did end up having a higher average percent transmittance at the 14 degrees Celsius. Oddly enough, the spinach was it did the best at this warmer room temperature setting than the cilantro when compared. And the cilantro ended up having the best transmittance out of the warmest temperature group, which makes kind of more sense when you think about that the cilantro is very steady, and the spinach had a few more spikes, ups and downs. It wasn't as stable. It's more suited for the cold temperatures, but maybe, depending on its growing situation, wasn't suited for specific things in specific temperatures. Now, the reason why this is important is because we're going through a time where the temperatures are fluctuating towards warmer in a lot of areas. And more importantly to the Midwest area where I live, is that it's expected that in the next couple of decades, corn is going to be more suited to being grown up in Canada than here in the Midwest because everything's going to shift up. Now that could cause a lot of livelihood problems for the farmers who rely on these acres upon acres of corn. So when we study stuff like this, we're kind of figuring out where is the tipping point? When do we have to start to adjust for these types of things? It's also becoming a problem in California where this change in temperature is creating this increase in natural disasters. In forest fires, in flooding, it's all happening more, and there seems to be a correlation or a causation with this temperature change. Now, for natural disasters, with most crop plants, you can just take the loss for one season and then you just replant. But this is becoming an issue for one of its biggest products there in California, almond orchards. Because with an orchard, if you lose it, you lose up to like 15 years of progress on those trees. It could take years for them to grow back. Meaning a lot of these farmers are having to switch livelihoods because of this. Because it's not worth the risk of having the orchard versus the reward they get back from it. And so if we start studying stuff like this now, maybe we can study how to adapt to these temperature changes and make it so that the growing season can be more planned out and we can optimize what plants are being grown where in the US. Thank you so much for listening to my project.